All right, hi everybody. Welcome to REEF for our April Fish and Friends evening. My name is Jill Keenert and I'm the director here at the Key Largo campus. Is that what I sound like? I'm the director of the Key Largo campus of REEF Environmental Education Foundation, or REEF for short. We are a nonprofit organization dedicated to marine conservation and helping people understand, explore, and protect our precious ocean environments. One of the ways we do that is by bringing speakers in here to our campus once a month this time of year. And we've got a great speaker for you, Anna, who is going to introduce herself, but is well known actually to many people here at REEF for various reasons, which I hope you'll go into. But she's legendary. I'll just say that around here. Everybody, lots of people online, I'm sure, are here, especially to hear her talk. And I know she's got some friends here in the audience as well. So just a few announcements before we get into that. If I could have the next slide from if I could have the next. Okay, there we go. Our Into the Blue book club is Thursday, April 11th. That's 2 days from now. Yay. All right. We got some fans for that here too. The book is Many Things Under a Rock, The Mysteries of Octopuses. And it's incredible is the word from the second row. I personally haven't read the book, and you don't have to have read the book to participate in this online book club webinar. But you can find information about it on our website at reef.org. We're also having a lionfish jewelry workshop here in this room next Wednesday from 6 to 8 p.m. This is where we take the spines or the fins from lionfish and make amazing jewelry out of them. And I've seen a couple of people here already with the lionfish jewelry. So that workshop is available for registration on our website as well. Next slide. But the big thing coming up here in our community is our Arts and Science Festival. This is the third year, Moose? Fourth year? Second year. Getting a little ahead of myself. For this festival is really unique in terms of combining art and science. And I've heard Moose say before that lots of people think you have to be an expert to be a scientist. Lots of people think you have to be an expert to be an artist. What we really want to do is make both of those things accessible and accessible together in the Art and Science Festival. So that's free to the public. There'll be lots of educational and fun activities. We've got vendors coming from around the community, also family oriented, to help us make that connection for the Art and Science Festival. So I hope you'll join us for that on the 27th. Next. I want to say thank you to our volunteers who help make evenings like this possible, and to our interns who are also still out there helping to clean up. I'm going to turn it to community announcements, but I know we have one community announcement in particular, which is that it's Bob's birthday today. So I thought we should all sing happy birthday to him. All right. Happy birthday to you. Happy birthday to you. Happy birthday, dear Bob. Happy birthday to you. Great. Yay. Now, hopefully this will be in one of your memorable birthdays of your life. But are there any, seriously, folks, any other community announcements for marine science, conservation, other birthdays might be around? No, okay. Everyone online is freaked out about that. Sorry, guys. This is what we do. It's a small town. All right, next slide. Want to say thank you to the funders. As I mentioned, part of the reason why we want the information for the sign up is to help show our thanks to grant agreements from the Florida Department of Environmental Protection, Florida Coastal Management Program, and the Office for Coastal Management for support for these kinds of programs. And nothing you will hear tonight is directly reflective of any of them, as you can read. But this is really important work and important support for our work that we're proud to share. And then the next one is back to Anna. All right, let's go. She's got about 50 minutes of stuff to talk about. We want to try to get ourselves out of here at 8. So that's our plan. Thank you.
Hi, everyone. Thank you so much for having me. My name is Anna, and I was a REEF intern. Yes, it was the beginning of a very great and still ongoing career. But I started in fall of 2014, which means I am now approaching 10 years since I was a REEF intern. And I enjoyed my time here so much that I had to come back to the Keys. Predominantly, I worked on the lionfish, invasive lionfish threshold capacity research with Stephanie Green. So that was a big focus of my time as an intern here. So fall of 2014 and spring of 2015. And so today, sorry, it's a little bit of a double slide, but I will be talking about the work that my office does monitoring coastal Everglades ecosystems. We do this by looking at fish and many other things beyond that. I am a fish person, carry that over from my reef stuff. So I'll definitely be talking a lot about that work that we do, but also the other work with Audubon's Everglades Science Center. So our story starts off in 1939. So I got to build a little context here. At this point in time, Florida and America are going through the Great Depression. Florida's land boom that was really popping off in the 1920s and was thriving and really holding up the market totally collapsed, in part because of 2 devastating hurricanes that happened here in Florida. As part of the New Deal, this is also when we start to see the conversion of Flagler Railroad into overseas highway. That work was completed in 1938. So I thought that was a pretty fun factoid to include, considering how iconic both of those things are. And at this point, we now have 2 refuges in the Lower Keys dedicated to protecting migratory and water bird species. So in 1908, we had the creation of the Key West Wildlife Refuge here, National Wildlife Refuge. And then 1938, we have the Great White Heron National Wildlife Refuge. And those were created in a direct response to the threats that birds were facing in the late 1800s, which was hunting due to the pluming trade. It was the height of fashion, especially in women's fashion, for clothing, hats, articles to be adorned with these very ornate feather pieces. And the demand was so high that they estimate about 71% of Florida's water bird population was eliminated in 30 years, from 1880 to the 1910s. So the creation of these 2 refuges were critical to preserving those birds and the land that they depended on, land and water habitats that they depended on for breeding and nesting. So at this point in time, in 1939, plume hunting had been illegal in the state of Florida for about 40 years. In 1901, the Audubon Model Law was passed by the Florida legislature, eliminating or making it illegal to harvest birds for feathers. And in 1918, we see that go onto a national level with the passing of the Migratory Bird Treaty Act, which was essentially doing that same thing, making plume hunting and hunting of birds outside of designated game seasons and game species illegal. So we're starting to see this rejuvenation and this revival of water bird species in Florida Bay, and the rookeries come back to life, except for one particular bird, the roseate spoonbill, which has become our office's flagship species. And that's where our story begins, because for some reason, while all of these other bird populations were rebounding, the roseate spoonbill was not. And people in the Audubon wanted to figure out why. At that point in time, it's estimated that the breeding population consisted of 5 nests total, all located here on this key, which then was called Bottle Point Key. But if you are familiar with Bottle Key, that's what we're talking about. It's just north of Tavernier here in Plantation Key. So the National Audubon Society's director sent Robert Porter Allen to Tavernier with his family to figure out what was going on. But unfortunately, he was immediately faced with a challenge. Traditionally, in these times, ornithology studies And learning more about birds involved killing them and examining their stomach contents to figure out what they ate, how they ate, when they ate, so on and so forth. That was not possible with the roseate spoonbill populations. There were so few of them that if that was the method in which Robert Porter Allen went with, the study would have been over very, very quickly. It wouldn't have been much of a population to study. So he really shifts to an observation form of studying these birds, which led to the creation of his office, at the northern point of Bottle Point Key. The breeding colony was on the southern end of the key. And so by creating this office, he was able to camp out at this island for weeks at a time and really observe what these species or what these individuals were doing during nesting season. As someone who goes out there like every other week, I don't know how he did it, to be honest. It is really buggy and certainly tests my mettle. So kudos to this guy. So that first study period went from October of 1939 to April of 1940. Pops over to Texas to study the roseate spoonbill populations there. Goes up to New York to do some literary research. Makes his way back down to the Keys. That's a close-up of Bottle Key. Sorry about that. And camps out at Low Key here. Right now, at this point in time, spends 5 months focusing on feeding and foraging, and then loads up that camp onto this 18-foot skiff he calls the Croc, and does the last few months of following and studying roseate spoonbills along the southwest coast of Florida. So after a cumulative 16 months of study over about 2 years, Robert Porter Allen becomes an expert on these birds. He observed their plumage changes throughout their juvenile to their mature sexual age, has notes and graphics filled with just behaviors of these birds during foraging, during nesting season, and predominantly learns one of the big discoveries is that roseate spoonbills feed on fish, not on mollusks, as previously believed. And part of the reason why they were struggling to recover was because of locals in the Keys killing the birds and harvesting their eggs for food during the Great Depression. Thankfully, the roseate spoonbill population starts to stabilize in the 1940s. You'll see me use this abbreviation here, ROSP, ROSP for short. That's the abbreviation that we use for this bird species in particular. It's the first 2 letters of roseate and then spoonbill. Smush them together. And Robert Porter Allen ends up becoming a bit of an icon. He has keys named after him in Florida Bay, the Bob Allen Keys. And he becomes a very big advocate for including Florida Bay into the designated protection areas of Everglades National Park. So very, very iconic figure, goes on to study whooping cranes, bald eagles, wood storks, very prominent ornithologist. And honestly, he says it best here. No snail that crawled, no worm that squirmed, no leaf that grew would escape my ardent attentions. Like, this man was zoned in. And honestly, it's very, very commendable. So that leads us to our office and the work that we do. The Everglades Science Center has been on the forefront of Everglades monitoring and research since 1939. We're one of the longest running Audubon offices. Actually, and our current mission is to restore freshwater flows to Florida Bay through the Everglades National Park to improve conditions for roseate spoonbills and other wading birds by reversing the effects of harmful flood control and water supply projects, which is a pretty daunting undertaking considering the amount of changes that the Everglades have gone through. And I'll kind of talk about that in the next few slides. But here are basically our 4 main projects that we do. We have our hydrologic monitoring project, examining water quality throughout the Everglades in multiple spots throughout the hydraulic year. Subaquatic vegetation surveys. We still continue to do roseate spoonbill monitoring to this day. And also the prey, I'm saying it out loud, at the prey-based fish monitoring program. Basically seeing what food is available for our spoonbills. So some quick rundown on Everglades ecology. It is truly a unique environment that's unlike anywhere else in the world. Part of that uniqueness comes from the fact that the Everglades' source of water is through precipitation, unlike many other aquatic systems which have a headwater. Florida's hydrologic year is marked by a wet and a dry season, and that rainy season is when we get that maximum input of fresh water supply, creating sheet flow. There's a minute decrease in elevation from the north part of the system down to Florida Bay. So as water enters the system, it slowly migrates its way down this elevation, creating sheet flow and giving the Everglades its nickname, River of Grass. That eventually ends up in Florida Bay. Florida Bay is the final ending stage of this ecosystem. And honestly, it is a biodiversity hotspot. There's a diverse amount of land within the Everglades itself, all changing just based on minute changes in elevation. It's at the nexus of multiple ocean currents from the Atlantic, Caribbean, and Gulf, and it's sandwiched right in between the temperate and tropical zones, making it a subtropical zone that's got a cool mix of both of those features. One iconic species of the tropic zones that we get up here is the West Indian mahogany tree. So a very unique place. Marjorie Stoneman Douglas also said it best. There are no other Everglades in the world. They have always been one of the unique regions of Earth. Remote, never wholly known, nothing anywhere else is like them. And unfortunately, this ecosystem is greatly imperiled and faces quite a few threats. The creation of the Everglades National Park has created a bit of a slowdown on the loss of habitat, but nonetheless, only 50% of the Everglades remain. And there has been a significant flow or significant efforts to alter the water flow. 70% of the historic water flow in the Everglades is gone. And so a lot of that was due to the change of land into agriculture and urban development. Both of the loss of habitat and altered water flow have led to poor water quality. So we can see that normally this immense system that took miles and miles to kind of filter out has been really streamlined and narrowed into a series of canals with a very significantly less amount of water moving through. That means water has less time to filter. These canals kind of straight shoot the water into these marine ecosystems, and the altered water flow, specifically through Lake St. Lucie and the Caloosahatchee River, result in a lot of harmful algal blooms for those specific estuaries. Seawater rise and saltwater inundation and sea level rise have also become a very big threat to the Everglades. It was a historically freshwater system that throughout time and the altering of the water flow has become significantly saltier. And where we do a lot of our work, sorry about that, those environments have become very fluctuating in regards to salinity. So during the wet season, they can be incredibly fresh due to the input of fresh water, but during the dry season can become incredibly salty. So we're losing that stability in these marine ecosystems as well. And of course, invasive species are always a problem down here. And the big one that we currently face are these Mayan cichlids that were introduced into our freshwater systems. All right, so now onto our projects. We, to this day, continue to monitor roseate spoonbill nesting in Florida Bay and the Everglades and determine the success of those birds each season. They traditionally nest from November to May, but current trends are showing that as sea levels and water levels are taking longer to recede as we enter that dry season, that it's actually shifting into the start of that season, shifting into December. Our bird person likes to say that her first nest she calls her Christmas nests because that's around the time she finds them now, since there's been this delayed onset due to that decrease or that water not drawing down. All of these red dots indicate places that were historic colonies. For ease of monitoring and checking, Bird Crew splits them up into 5 separate regions of Northwest, Southwest, Central, Southeast, and Northeast. And those regions kind of correlate with the historic foraging grounds of our fish research sites here, because all of our fish research sites have been built because those were areas where spoonbills have been seen foraging for food. The success of roseate spoonbill nesting is dependent on the water levels in the Everglades. So as I mentioned earlier, we have a wet and a dry season. And during that wet season, it's a moment of high reproduction, and these fish disperse all across the Everglades or those coastal Everglades systems. More water means that they are able to kind of hang out in the areas that are traditionally shallower and deeper, and there's just a lot of reproduction going on and a lot of new individuals being added to the population. As the dry season happens, those water levels dry down because we're not getting any rainfall, and the fish start to aggregate in these deeper parts of the water. And this is critical for spoonbill success. They are tactile feeders. So these drawdown events create an abundance of food in a very small area. And we specifically look for this 13-centimeter water depth drawdown, because that is like the first concentration event. And if water levels drop down to 13 centimeters, that means that spoonbills are able to forage more successfully for food to feed their chicks. Unfortunately, what we're seeing now is it's getting increasingly harder to achieve this drawdown for a significant amount of time to truly be helpful for our spoonbills. It needs to be, water levels need to be at 13 centimeters for about 50 days to really ensure like really good success for these nests. So when our bird team goes out and looks at these nests, they're keeping track and recording the number of eggs laying in nests, the total number of eggs that hatch, and the total amount of chicks that survive to fledgling age, which is about 21 days. Incubation for eggs are about 20 to 22 days long. These chicks here are what we call stage 1, late stage 1 chicks. It's kind of hard to like look at a chick and be like, that chick is 5 days old for sure. So we separate them into age stages essentially. So stage 1 means that these chicks are about 0 to 7 days old. And according to the bird person, I'm a fish girl, not a bird, not too much of a bird person. And so these are late stage. So we're talking maybe 5 to 7 days. With these ones here being stage 2, so they're 9 to 13 days old. Here is a cute video of some freshly hatched chicks. That one in the middle is like literally 20 minutes old. The tech observed that nest, saw that there were the 2 day-old, 1 to 2 day-old chicks on the bottom and the top, and that chick was an egg, came back 20 minutes later, and there was a little baby. So fresh little roseate spoonbill there. You can tell they're very, very freshly hatched because their eyes don't open. And they don't have their iconic spoonbill until they reach more of that stage 3 fledgling age, which we can see this chick starting to get here with that more spoon-like bill. Bird work is pretty arduous work in some pretty tough environments. Certainly not a place I would do well in. I'm quite klutzy. Luckily, when I fall, I just fall into water, not into cacti. And there's a lot of climbing that's involved too. If birds' nests can't be seen through climbing though, they're too high like this one, mirror poles are used. Very long extendable poles with basically like those security mirrors you see in like gas stations and stuff on the edge, on the end of it, so that way they can count the eggs, chicks, all of that stuff. We have elevated our project to include more modern technology. We can only, due to manpower and resources, check these regions like every 2 weeks. So that's a lot of time where things happen and we don't know whether the chicks are being predated on, what is predating on them, if they're hatching, so on and so forth. So our trail cam project allows us to really observe these birds throughout this nesting project. And so here we can, if you can see, we have one bird tech installing the nest or the trail cam there. We have another one hiding behind these mangroves here. Ideal setup is to have the trail camera facing the nest with an unobstructed view, no leaves, no branches. You want a nice, perfect view inside the nest. And here's our bird person doing all that hard work, climbing up, making sure those angles are good. And this is also useful because it reduces human impact on the nest. If a big giant ape thing was climbing up and checking out those chicks every day, that would impact the parents coming to the nest, possibly stress out both the parents and the chicks. So it's a good mix of us being able to see a lot of things happen while not impacting the birds too much. So here we have a good angle of a roseate spoonbill pair and their nest with some eggs. And this is just a superb view of what's going on in the nest. So these chicks are about stage 2 as well, estimated about 9 days, but that's 7 to 13 age range. Unfortunately, what we are seeing is that Florida Bay is not really a successful nesting area for these birds anymore. They are starting to migrate farther and farther north when 2 areas where that 13 centimeter threshold can be achieved. And so we can see back in the 40s or 30s, 40s, and 50s, they really hung out on that on those keys closest to the island at that time. That was when a lot of the mangrove and foraging sites on the keys were still thriving before they got converted into neighborhoods and drained. And then we can just start seeing that they really start shifting around with kind of this 2018, 2019 nesting season, really migrating up into the Everglades itself. And just overall a trend that, you know, they were doing pretty well in the 70s, but just overall their nesting success has dropped over time. And the number of nests overall have dropped over time. One interesting thing we are seeing, though, is that they're starting to forage on keys in Florida Bay, when traditionally that was where they nested and then migrated around to, whether it was the coastal Everglades here or the Keys. So very, very interesting. Thankfully for the population as a whole, they are a highly mobile and adaptable species. So even if they're not doing well here in Florida Bay, they're looking, they're going out, they're moving around and finding areas that might be a bit more conducive for nest success. And so that's where our satellite tracking project comes in, to not only track their movements during nesting season to see where they forage, where their new food supplies are, but to also just see where they're going throughout the rest of the year. And so we've got a video of a bird here really making its way around Florida Bay and the Everglades. Yeah, they've got some pretty erratic movements. And we're going to see a really big jump very soon. Still hanging out. And then boom. Apparently these water conservation areas are becoming a popular nesting site for these birds. Those water levels are really drawing down in that concentrated way to really allow them to be successful. And I don't know what was in the water right here, but he really dug the Melrose area. And so, you know, sometimes they'll have just that like erratic back and forth movement to a central area, coming all the way back down. And this can really kind of help us also identify where the nests are. If they kind of keep returning to one place and then all their movements radiate out of that area, that can help us pinpoint which key and like where exactly on the island their nests are. That is just one bird. And I believe that time frame is over the course of a year. Yeah, so some birds, they park it out in Florida Bay and they stay there. Others are migrating across the state. Some have even gone to Georgia. So once again, like really mobile population. Not great for us if we want to see them nesting successfully here, but overall, like promising for the bird. They will find a way. And as we've kind of heard, or as I've kind of talked about, the accessibility of the prey for these birds is really what makes or breaks a successful nesting season. And so we have to take a look at the fish populations to really see what is available for these birds. And this is also important because fish is the most abundant food source for all organisms, all predators in the Everglades. We estimate the identity, or we identify the species and estimate their biomass, their density, and their availability. And we have 12 sites ranging from Turkey Point all the way down to 2 Cape Sable sites where we examine and we collect fish and try to determine what is available for these spoonbills. So each site is sampled 8 months out of the year. We have our predominant sampling season during November and from November to April. We have 2 basically control months in the rainy season in June and September to see how fish populations differ throughout those 2 seasons. There are 6 nets per site. We have 3 creek nets, which are, for example, these deeper nets that we see here in that water level image. That deeper area is what we would call the creek nets, and then flats nets. So as water levels draw down, water levels disappear and decrease in those areas. Our nets are created by, they're 9x9 squares with PVC pipes anchoring each corner. There is a rebar frame, and that rebar frame is what we attach these nets to. It is a process. Our fish samples are actually 3 days long, day 1 being set up and kind of making sure that the nets are ready to deploy. For day 2, our sample day, where they look like this, and then day 3, where we break them down. There used to be 9 nets. Don't know how people before me did all of that, because 6 nets is already more than enough for me. But yeah, this is an example of our flats nets. And when water levels get low enough, we can't take our kayaks around the net. So we have to basically use a long piece of wood with some iron bars on the bottom and kind of throw them. We call them our throwables. Throw them down so that way we don't disturb the environment. Because obviously there's a certain level of disruption that happens with this process, but we really want to minimize it to just specifically these nests, or these, nets. If you are a person who's a little prone to motion sickness, this is kind of like a high-speed video of just a very small portion of our journey to site, so be mindful of that. But this is a path we call the Long Narrows that we use on a to get to Taylor River. As the name implies, it's long, very twisty, and very narrow. So this is just like a short portion of our trip up to Taylor River. As a newbie, it was very much like Mario Kart with the bumpers on. A lot of ping-ponging back and forth, but got the hang of it eventually. So our sites, we access them through bay boats and john boats. Here you can see our boat set up for one of our Everglades National Park sites, 6 nets, 2 kayaks, all sorts of stuff in the back to get the work done. Here is kind of our setup for our john boats, very much like Tetris. When it comes to setting up the nets, day 1 is ensuring that these nets are rolled up on the rebar. The idea is we don't want these nets touching the water because that can disrupt fish behaviors if something's dipping and moving around because of wind, especially in the dry season. That's when we get a lot of high wind and bit rougher weather. And we prop up the rebar with nails that are tied to these lines here. We call these our pull strings. And the goal is that on the morning of day 2, we can pull those strings and the net will drop. Each site has a hydro station, so we are also responsible for collecting physical data to just quality check the data that our hydro stations send to us via satellite. So on sample day, we have 3 hours after sunrise to drop those nets. After that 3-hour mark, that's when the sun's angle and shadows kind of also impact fish behavior. So we really need to be out there on the water by first light to ensure that these nets drop. All of our work for our fish sites is done from a kayak. So also probably some videos that I wish my coworkers, they wish they took of me trying to successfully pull nets for the first few tries. Really overshot it, fell off my kayak quite a few times. But we drop the nets. Those strings have the nails attached, pull those out. The rebar frame drops down. We ensure that the nets and the rebar are flush to the bottom. We do not stand in our sites like this guy is doing. That was way, way, way back in the day. But essentially, we do collect the fish using rotenone, which is a drug that is used to inhibit a fish's gills from absorbing oxygen. So the idea is they come to the surface to get air, and that's when we collect them with our dip nets here and put them in baggies to identify in the lab. Rotenone is we neutralize it with another chemical called potassium rotenone, so that way it doesn't leach through the net and impact the surrounding environment. We're really just trying to make sure the water that we dose stays in the net, and we sample for about 30 minutes. So after 30 minutes, if we don't see any fish activity, that's when we add the rotenone and move on to the next nets. Day 3, takedown day. Here's a fun video of me kind of rolling up those nets. We're talking 50+ feet of Gore-Tex fabric that we have to roll up nice and neat. Otherwise, it's a severe tripping hazard. So there's, here's the process of us kind of like breaking down that net, and it rolled up on my kayak before I roll it up on the boat. After that, we make sure that everything's properly labeled so that way the fish processing can commence. So this is the point where we ID the fish, we weigh them, we get total length and standard length. And those numbers are used to help us get biomass and assess that availability at all of these sites. So we got quite a mix of fish species here. This big one, actually almost all of these ones here are those invasive mine cichlids. They are doing quite well at a lot of these sites, unfortunately. But we've got some more fresh or salty water fish here, our moharas. And these look like, I had to say, those might be our sailfin mollies. So we have quite a nice mix, and we see a lot of different fish species out there. Sailfin mollies are important because they're a really significant part of the spoonbill's diet. And this is a cool melanistic one that's got that cool modeling color. These are kind of oglohaling fish, so they can handle that salinity fluctuation. This is a pike killifish, which is predominantly a freshwater fish. And then some other cool fish species, including the hog choker and the red drum. Yeah, I wish I could tell you why. I do not know. So some more of our projects include hydrology. Water quality impacts the species we see at these locations. It's really, indicative and important to monitor. So salinity, depth, and temperature are just some things we look at throughout the whole hydro year. These are satellite based, so they can collect the collect the data with a sensor probe that's here, gets sent to the box, and at the top of the hour, every day, it is then sent to our office. So all of our fish sites have these hydro stations, but we also started installing them on some keys because of that shift we've seen with spoonbills foraging on keys themselves. So we definitely want to get in there and kind of see what's going on with the water quality there. And then tied with that is subaquatic vegetation survey. So this is the other predominant project I work on. At every fish site, we have a transect that we use to basically observe any changes in plant composition. We know those changes are happening because these freshwater systems are becoming more saltwater. So we want to monitor those changes and be able to look into the data and see how that works. So over here is bladderwort, which is actually a flowering algae, and that is a very freshwater species. It requires some pretty stable salinity levels in order to really establish, and all of this is done through snorkeling. So we use the point intercept coverage technique. Essentially, we have a quadrant with 5 points. You can kind of see that there are some strings tied throughout here. We throw that quadrant out, and if there is a plant species touching one of those points, we identify it and we record it. And those numbers are then used to calculate the estimated percent coverage for those sites. So at a specific site, we have this one right here, which is Taylor River, which is the twisty video that I had earlier. This is where our fish site happens, and that is where our first SAV transect is taken. And then all of these are sub-sites within our Taylor River SAV transect. So it runs from freshwater down to saltwater. We want to see those changes in plant composition as we make our way into those more saline environments. So each spot is a fixed location with a randomization added by the random 12 throws, whether they happen in a circle around that fixed point or 6 throws out and 6 throws back. We have that mix of standard and fixed as well as randomized. And what we're, oh, sorry, that got a little carried away. So basically what we're seeing here is now we're going to dive into the results of Taylor River and what we've noticed with water levels, salinity, plants, and fish in the hydro year of 2022 to 2023. So that hydro year starts in June, ends in the following May year. So sloughs are deeper bodies of water that carry fresh water through the Everglades into Florida Bay. Shark River Slough and Taylor River are kind of the two big ones. Shark River Slough, I guess, being like the most intact in a sense. But what we've seen is that we had record high water levels this year at the beginning of the hydro season. I'm going to pop over here. At the beginning of the hydro year. So high levels, and we had this massive spike of water levels due to Hurricane Ian. This white line are the trends for the hydro year 2022-2023, with this gray mark kind of being the average, and this line on top being the maximum water levels and the minimum water levels below. The red line is that 13 centimeter threshold that we're kind of aiming for. So as we can see, really, really high levels of water, even leading into the beginning of spoonbill nesting season in November. There was a tiny moment right here where we got the water levels below 13 centimeters. But we need that sustained over a period of like at least 50 days for optimum success. And we got it for a fraction. So certainly not enough of a time for spoonbill nesting success. Popping back over. Salinity wise, we can see that while there were some periods of stability with very low salinity, which is good. Stability means plants have, those freshwater plants and fish species have time to establish. We did have spikes of salinity throughout the dry season. So that lack of stability doesn't really bode well for those fish and plant species. Overall, the average plant cover in Taylor River Slough, which was that transect that I was pointing out before, dropped. We do have the establishment of Utricularia, which is that bladderwort flowering algae, which does need that fresh water for a long period of time to establish, but definitely something that we want to keep an eye on. And overall, fish-wise, it was actually a decent year. The restoration goal for fish populations is to have about 40% of the fish be freshwater fish, and we hit 48. So we want to see species like largemouth bass, our black acara, spotted tilapia, and that bluefin killifish, which was kind of the very narrow pointy guy a few slides back. But we also caught less fish than normal. So kind of another thing to keep an eye on. And here we can see these species in red. Those are the low salinity fish, which include the crested goby, which I remember starting this job and doing SAV, and I was like, wait a second, like, what is that guy doing back here? But they actually go pretty far back into the coastal Everglades where we have our sites. And then the gold spotted killifish being one of those high salinity fish that has been caught at these sites. So all in all of our work has its challenges. I've been on quite a few broken down boats with need of rescue. This one was in Long Sound with our John boat. But I've also gotten stuck in Lake Ingram. And yeah, you know, it's a good time. Certainly makes you wonder. And our bird people certainly do a lot of insane and dirty work. And, you know, there's always the fun random thunderstorm that pops up in the in those summer months that we have to keep an eye out for as well. But we do get to see some cool things like croc encounters. No crocs were harmed in this video. It was in fact retrieved. You know, I went to do SAV and just like throw my crocs out of the way, and my luck meant that that croc bounced and fell right out of the boat. So thankfully my friend was in the water to grab that for me. But we do get to see the real deal as well sometimes too, from these big adults to even our little juvenile guys. And there are lots of other cool critters we get to see. You guys may have heard how flamingos have kind of been hanging out at Snakebite. So that's some pretty exciting news. But water snakes, dolphins at some of our very salty sites, specifically Turkey Point, conical sea stars, and, you know, froggies and crabs. So all sorts of cool things that we get to see beyond just the fish and the birds, which are pretty cool themselves. So of course, got to thank the team. It's a very small crew, but we all pull through and do all this amazing work and get it done, and our sponsors who help fund our work as well. So I will open it up for questions if there are any. Sorry, I know that was a lot of information thrown out. Thank you.
Great. Thank you so much, Anna.
Of course.
Questions? Comments? Moose has a question. Okay. Chick. Mita's got one. Just repeat the.
Documented preying on the vine cichlids, or are they still not really recognized?
So he was asking if the spoonbills have been observed preying on Mayan cichlids. And to be honest, we don't really get that information unless, you know, an individual has died, or if there's like a regurgitation event, essentially in the process of trapping the birds and banding them, they will regurgitate their food. That is something that the bird crew tries really hard not to do. But theoretically, as long as the bird is smaller than 6.5 centimeters, it can fit in its beak. So they're pretty indiscriminate. They're tactile feeders. If it's something that touches their beak, they're gonna try and catch it. So there really wouldn't be any reason to assume that they wouldn't eat those fish. It's just the problem is that those Mayan cichlids have become so established that they can get really, really big. But, you know, I think we find that or when I looked it up once, that they do a lot of breeding in like April and May. So there are some babies that pop out, but once again, you know, there's really not anything controlling their population. So very similar to the lionfish, if they get too big, they can really have that like cascading population effect of eating the smaller guys that the spoonbills rely on. Yes.
So we're doing a lot of Everglades restoration.
Mhm.
What are you expecting to see from that, and have you seen any results yet?
All right. So the question was, we've been seeing a lot of Everglades restoration. What are we expecting to see, and what have we seen so far? So this is where our work becomes really critical, because we take this data and we analyze it and we send it to the Army Corps of Engineers, to South Florida Water Management District, to let them know what is good water management and what is bad water management. And unfortunately, it seems like they're just kind of letting water out willy-nilly. There's not really a system to kind of emulate that historic water flow. I think a release just happened a few weeks ago, traditionally in a time where we don't have this water. We're supposed to get that drawdown. So that's what our main goal is, to really provide them the data of like, that you just did, the releasing water in the middle of March, not the best idea. Like, that's not really helping things.
There's a big slug of rain.
That's true. We do have to keep in mind that like a lot of this water management is happening because of the urbanization that has happened in the areas. Yeah. Ooh. So there's also a very important reason why that draining also needs to happen. So I guess also like giving them, you know, a course of action, just giving them the information of like, here's maybe what efforts can be done to like lower the waters without having too much of an impact. We do see that we are getting a bit closer to these drawdown events with kind of the information that we've been giving them. So, you know, unfortunately, we're never going to get that historic flow. We are too far gone into the altered system to really achieve it. But if we can provide them information to get closer to it within the means that is necessary, then that's great. But we have been seeing, you know, trends toward more drawdowns in the dry season, traditionally when we're supposed to have them. Yes.
Maybe 3 or 4 years ago, I heard a gentleman speaking, a researcher, the roseate spoonbills. And my recollection is he was lamenting that they were retreating up to the southern Everglades and not nesting out here successfully in Florida Bay. And my recollection is that he was saying that it was because that the young were so dependent on a particular type of food that could only be found at that certain salinity level. And that's why they had to keep moving further
Mm.
north because that salinity was not here. But my question has to do then with where we saw that bird flying around all over. That bird was going up into areas where I would assume that that very particular requirement would not necessarily be met. And I wonder, was he overstating it? Was I misunderstanding it? Or?
That could have been the research at the time. So the question is that a few years ago, a researcher spoke about how birds are migrating in the southern part of the Everglades, not really nesting here in Florida Bay because of salinity. And there's a specific level of salinity that was needed. And I do think that like plays a part in it. What we're learning now is it really is that water level and the impacts of sea level rise impacting the success of the nests. I think traditionally, traditionally these areas did have freshwater species. So that inundation of salt water is having an impact on on fish communities. They're changing over time because of that salinity. But spoonbills are pretty indiscriminate. If it fits within their beaks, that 6.5 centimeter limit, then they can eat it. And really, ultimately, it's the water levels themselves that are becoming the problem and are the problem just because that drawdown isn't happening. So there's just not that concentration of fish in those smaller areas to really allow the spoonbills to get enough food to feed their chicks so that way they can be successful. So I'm not sure who that researcher was, and that could have been at the time kind of the leading thought. But I am gonna ask around about that and see maybe if like salinity has a bigger part to play than previously believed.
It's a very specialized type of food that the young required.
Okay.
And so
It's definitely fish. So at the time, it could have been that just those changing communities can impact the sailfin molly being like their kind of main staple. Those are like the low salinity freshwater fish. So it's possible that he could have been referencing something more like the fact that their food itself is changing. But I'm intrigued. I will ask around. Any other questions? Yeah.
Yeah, do they reuse their nests?
Oh, that is an excellent question. And I actually am not sure. I asked all the bird people questions and I was like, what are they going to hit me with? And they're like, they're going to ask you this and they're going to ask you this. And I was like, because I don't do much of the bird. I actually did 3 days of bird work. And I was like, get me back in those kayaks and never again. So I will have to ask. Yes. Yeah, there's right now the estimates are like a 50-50 shot of success. So it sounded like, because I asked, about 150 breeding pairs kind of like in Florida Bay at the moment. So it's definitely just like not as great as it used to be for them. Oh, nesting, just like traditionally from November to April, but that is shifting more from December to end of April, May almost. It's almost like taking a shift back because of higher water levels at the beginning of the season. Yeah, you got another one?
Well, I have one question and one comment. First, I'll make my comment that last year, a spoonbill was spotted in the Anacostia River up in Washington, D.C. Well, I have that up there, but and I don't know whether it's having fun or not.
Who's to say?
My question has to do with pythons in the Everglades. Have you seen pythons while you're out there, and any indication that they're affecting the bird population?
Have I seen pythons out there, and are they affecting the bird population? I can't give you any information on the bird population, but I've had a coworker like wrangle a python before. So I think that was like West Joe Bay. So in the west part of Joe Bay. So yeah, they're out there and like we can see the tracks as like we make our way. That site in particular has a very like narrow creek that one person pushes the John boat through and the other person follows in a kayak. And for the longest time, I was like, wow, these are some tiny little like alligator like marks coming through the sawgrass and the Eliocharis, but actually like those are python paths. So they're there. The only snakes I've really seen out there have been water snakes and water moccasins. But yeah, my mom I'm the only biologist in the family. So my mom's like, you better text me when you get home every day if you're in the field. Cause she's like, the crocs, the sharks, all of it. She's like, nope, you need to let me know. So that's all.
That's amazing. Anna, thank you so much. I can't let you go without number one saying how proud we are of your success as a REEF intern alumni. And I wonder if you don't have just like one or two words to say to our current marine conservation interns and fellows, word of advice or encouragement for them at this point in their semester with us.
I mean, there's so much ahead, and just kind of a thing that has followed me through since I was in college was that, like science is science. So whether it's happening in the ocean, in the Everglades, with birds, in the Great Lakes, like if you love science, you really can apply that passion to any organism, to any species, to any ecosystem, because we just love being out there, and we love experiencing new things and working with the animals. So, just if passion takes you in a place you didn't expect before, like that can be a really cool and new exciting thing.
Thank you. Thank you very much. I've got one more slide. Thank you. Oh, okay. Is there an ending slide? Oh, okay.
Do you need me to go back to the front?
No. It's fine. We have a newsletter, a Keys community newsletter that I wanted you guys to sign up for. If you're not already signed up for our newsletter, send me a note, let me know, and we'll get you onto that. But again, thank you to Anna. Thank you to our volunteers and our interns and everyone else for being here. We'll see you in May for our next talk, if not sooner. Lionfish. Thanks, everybody.